Thank you. So it's a great pleasure to be here in beautiful Chicago, and I've now got the bay in the background. Okay, thank you to the organisers for inviting me to this very exciting meeting. So as Alex said, I'm going to talk about some of our work that I've been doing with Andrew McCainch in Warwick on human cell division. So there's some sub-themes here: Bayesian model fitting and the analysis of the collective, which I'll go into. Okay, so just to get us on the same page, mammalian cell division, the phase I'm interested in is this mechanical phase of mitosis. But firstly, I'd like to, of course, mention that in S phase, you duplicate your chromosomes, and at that point, they get stuck together. The duplicates are stuck together, giving you this familiar X shape. And so mitosis is split into phases. You've got nuclear envelope breakdown, and then the chromosomes, it's just the chromatids, this pair, float around and get captured by the spindle poles. You form the spindle, and then during congression, they are moved to the cell midplane, and during metaphase, the chromosomes undergo oscillations in practically most species. And then it goes into anaphase as you dissolve the glue between the two sister chromatids. So the spindle microtubule, composed of microtubules, connects to the chromosomes at the kinetochore, a macromolecular complex, which I will discuss. Okay, so this is the sort of data we have. This is a lattice light sheet. It's very gentle on the cell, so you can image for a long time. This is 3 to 4 frames, 2 colors, going over about 30 minutes. In green, we've got the kinetochores tagged on a molecule called NDC-80. NDC-80 is like a tip tracker of the microtubules. It binds to microtubules on the plus ends. In magenta, you've got tubulin. It's not good enough to see fibers or K fibers, but it does give you a guide on the context of the spindle, and we're able to just track the spindle poles. So first of all, I'm going to show you some of my work on metaphase. So the kinetochores undergo pseudoperiodic oscillations, which you may well see here going on. And this is what it looks like if you project along the metaphase plate your x coordinate. You see really good sawtooth oscillations. So there's a number of questions. What drives oscillations? Are all kinetochores equal? And are oscillations time homogeneous? So we've got near complete tracking. There's 92 kinetochores, and these are our tracks, practically 90. Okay, going all the way through, most of them going all the way through in this cell. And we compare them up, so we get about 46 pairs. And we have, for this data set, we had 26 RP1 cells. It's 2 seconds per frame, about 800 kinetochore pairs, which we analyzed. So the models we fit, and that's really what I'm going to discuss in this talk, about fitting mechanical models, is very simple. The reason it's simple is because the data can only inform you so much on what's going on. So we're restricted by the complexity we can actually get the data. Okay, so especially a force balance model, we've got centralizing forces, for example, the polar ejection force. The poles give out microtubules which collide with the chromosome arms, pushing them into the midplane. You've got this centromeric spring joining our 2 kinetochores, our sisters. And then you've got the K fiber, so a bundle of about 10 microtubules, which attaches to the kinetochore, and it can either be depolymerizing or polymerizing. So we fit this model. It's Bayesian, so you fit all the parameters together. This is our model. You've got the frame-to-frame estimated speed. You've got the K fiber velocity, which can be pushing or pulling, depending on the state. So it's a hidden Markov model. And then you've got the spring. We've allowed for the fact it can be twisted in 3D. Then you've got the polar ejection force, which was linearized around the midplane, and then Gaussian noise. So we did this 10 years ago. We fitted this sort of model. So you take your trajectory. What it gives out is really nice annotation, tells you which direction what the polymerization state of the K fibers is on each sister. And these are parameters: pulling forces, the centralizing force, pushing forces, spring. You get it all out. One of the things, though, which I emphasize, is the fact it annotates for you. A very, very valuable tool. Okay, so we took that model and we said, well, there must be more information than this simple model. And we looked at 17 different models. Okay. One of the things we looked at was time homogeneity, adding time dependence to all these parameters. I'm not going to talk about that. I thought I'd just talk about this bit here, whether you ask whether sisters are equal. Okay, are they dynamically equal? So one of the things we said is, well, maybe the K fibers are different. Maybe their strengths are different. Or maybe the noise, the diffusion is different. So you fit those models, and lo and behold, we found 25% were not symmetric. And if you look at the profile, practically all of them have the pulling force of the K fiber is different between the two sisters. Some have different pushing forces, and an occasional one or two have different noise. Now, the pulling force is a dominant force driving the oscillations. It's the strongest force by order of magnitude, bigger than the spring, bigger than the pushing force. So it's not a surprise that you pick up asymmetry in V minus, because that's the strongest force. So when we asked, well, is this a special population of asymmetric kinetochores, asymmetric sisters? Well, no. It looked like it was just fluctuations. There was no half spindle asymmetry. It was all so there I'm getting at the fact that the centrioles are different ages. It suggests a random effect. One of the pieces of evidence we looked at was we said, well, you've got pulling force differences between your two sisters. Supposing we just ask if that distribution is any different than any two random kinetochores, and it's not, it's identical. So it suggests that kinetochores are just variable, meaning kinetochores or the K fiber. So if you think about it carefully, if you've got a pair of sisters and one pulls stronger than the other on average, it's going to pull it over away from the midplane towards the spindle pole with the strongest kinetochore. And so this is just the positioning of the kinetochores. You see it's pulled to off center. So then you can think, well, in an asymmetric pair, we've got a strong kinetochore, strong pulling force, a weak kinetochore, weak pulling force. So these will be ordered laterally along the metaphase plate.
So, like in this model, because the motion is a balance of forces, how can you tell if it's like the centering force which is changing versus the pulling force which is changing? Wouldn't those give the same resulting motion?
No, the pulling force is changing as it switches side. The centralizing force wouldn't.
And so that's just an assumption that the back and forth is due to the changing of the pulling force, but the centering force changes, stays constant. That's
essentially centralizing force is just a wind.
Okay.
Yeah. So that's how you separate them out. Okay, so here we're looking at all the kinetochores of a sister pair, which are then going to descend to this daughter. I've blanked out all the others. So each kinetochore here had another sister, which I'm not showing. And this is the positioning. This is a strong kinetochores being put, they have a stronger pulling force towards it. These ones have a weaker one. And this positioning is retained right through anaphase. So if you go back to these questions, what drives oscillations? Well, it's microtubule dynamic instability. There's a regulating switch somehow, still not totally sorted out. Are all kinetochores equal? Well, no. Random fluctuations make them unequal. There's also spatial variation across the metaphase plate. Are oscillation time homogeneous? I didn't show you that, but no, they're not. If you're interested, it's on bioRxiv. Okay, so we have a powerful set of tools to understand kinetical dynamics in metaphase.
Just for the previous one, how many did you try? How much data do you need to make these determination? Like if you had a smaller pool of data, would you be able to still?
Well, we are doing individual kinetical pair trajectory analysis.
But a lot of it, right?
Yeah, so we had 800 pairs.
So if you had like 20, would you be able to make
much more difficult? Because of course you'd still be able to see that positioning, I suspect, but you get power in numbers. We're talking about more subtle effects. So for concluding something about individual kinetochores, we can conclude whether asymmetric or not, that sort of thing. There's enough information there. But if you want to build up the statistics of a whole cell, you've got to do complete tracking. Okay, so we want to move these techniques to prometaphase, a much more challenging area. So I said the kinetochore attaches your chromosomes to the microtubules. It does it in two ways. You've got lateral attachment through motors, dynein and SEMP-E. Dynein, which is one we're going to be interested in, moves you towards the spindle pole. You also get end-on attachment, which through this molecule is tracker NDC-80. And this is slow. This is an order of magnitude slower than these guys. And so there's a number of states. The one you want is this one up here, biorientation, one kinetochore attached to one pole, the other kinetochore attached to the other pole. They start out in this state here, no attachments, and they've got to go through a whole set of attachments. So you can get errors, both kinetochores attached to the same pole, that sort of thing. Those get corrected. So the kinetochore, this macromolecular complex, brokers and generates forces, but it also changes composition. A key event is end-on attachment. When end-on attachment happens, dynein leaves. So it's sitting on the kinetochore, pre-attachment, so able to do this lateral attachment. You get end-on attachment, dynein leaves, it strips, takes cargo with it, and it takes the checkpoint proteins. So MAD2, which I'm going to mention later, is a key checkpoint protein. If MAD2 is on any kinetochore, the cell will not go into anaphase. So you've got to strip MAD2 off all of them, which means you've got to get end-on attachment on all of them. Okay, so let's look at the data. So this is prometaphase. This is our number of spots in red. You can see it rises through this movie, settling again at around 90, and the number of pairs again is high in 40s. So this rise here, this rise is due to the fact we have tagged NDC-80. NDC-80, this green dots, is loaded. So if I show you on the movie, you'll see they're dimmer at the beginning, then they load. This is outer kinetochore assembly. NDC-80 is coming from the nuclear envelope, and then as the nuclear envelope dissolves, it loads onto kinetochores. That's partly why we see the number of spots increase. The other thing, which is very interesting, is looking at the dynamics. At this nuclear envelope breakdown, you see the kinetochores are highly dispersed, and then they go through this contraction, some of them doing very fast movements. We call that a contraction event. So let's just look at that again. Got this rapid movement there. Okay. So we quantify the shape of this collective of kinetochores by looking at the covariance matrix. Okay. So a covariance matrix has 3 eigenvalues. We've got a small one. We always have a small one. We always have a small one. So the collective is like an ellipsoid and it's squashed. So this narrow direction will become the direction of the metaphase plate normal. And these other directions are wider and they come in this contraction. I've drawn it like a donut. It's because you often see the spots organized around in a donut. I mentioned the fast events. So this is our 3 frame speed in light blue. You get these fast movements early on in contraction. Okay, so this is our tracks with dragon tails, and these are our tracks. Some of them go right through our movie over about 30 minutes. These are our pairs. We get some early pairs, which we're then able to analyze. We actually pair up later in metaphase, because that's where we get really good pairing. Okay, so this is some of our tracks, just illustrating some of our tracks here. You can see these pairs moving together. So with tracks, with pairs, you can now analyze mechanics. So this is our outer kinetochore assembly, the intensity of our spots. Outer kinetochore assembly is complete. This is the tension, average tension in our pairs, rising over about 400 seconds. This is the sister pair order. So the sisters give you vectors, the orientation, initially random, then they start organizing themselves like a pneumatic material and going parallel. So you can see this rapid rise in order. We've also got the spindle poles, contract the spindle poles, and so we can get the length and the orientation of the spindle axis. So I should have said here, when I'm looking at the coordinate system, that we call this the minimum spread plane, which is the plane of our donut, and we got the minimum spread normal is normal to it. So the minimum spread plane will become our metaphase plate. Okay, so you get pictures like this. You've got outer kinetochore assembly, you've got tension, you've got our pneumatic order here, and at the same time, this is we've got the spindle pole distance increasing, but we've also got the kinetical populations rotating. So on this one, it started out at 45 degrees. Our donut was 45 degrees like that. It rotated. And it's coincident, this rotation is coincident with establishing tension, which it must be. Okay, so once attached, the spindle forces reorganize the kinetochores through a series of mechanical phases. That's basically what I've shown you. And we think that this should be understandable from basic mechanics and the attachment kinetics. We can analyze this in two methods. We've got the analysis of the collective dynamics. Those features we can extract from the collective, phase them, and we've got about 20 cells at the moment, which we are proceeding to analyze. But you can also look at individual kinetochore dynamics, phasing and annotating of kinetochore trajectories. So annotating, like I did in metaphase, that's what I want to do. I want to pick out when dynamics is changing. And there we'll have about 2,000 tracks, of which about 20% will be paired. Okay, so as a collective, we'd have 4 features. These features, outer kinetochore assembly, contraction, tension, generation, pneumatic order. And what we'd like to do is find the timings of these, relative timings, how they move into each other. We've been doing quite a bit of work looking at time warping methods and also fitting parameterized models. It's not very robust, unfortunately, and that's our problem. So I'm going to switch to looking at individual kinetochore dynamics. Very similar to what I did in metaphase, but we're going to develop new models to do this. So we use a switching model. It's one way, so you can move one, state 2, state 3. We have k states. In our analysis, we have 9. And these have associated unique dynamics. what we're saying is a diffusion model, and each one, the reason you change state is one of either the noise or the applied force changes direction or strength. So the data will dictate how far it goes down this sequence, okay, how many states it wants to use. So we implemented that model, and this is trajectory 17. It's this blue one here, and this is our probability of switching. Okay, so you got very high probability of switching at about just after 100 seconds. Well, 90 seconds. No, it's about 100 seconds, then 110 seconds. They got another period of switching here. So to visualize this as clearly as possible, this plot, I think, is really useful. It is the probability of being in a different state at time t2 from t1. So it just tells you whether you switched. So it looks like you see a lot of biological images looking like this. It looks like high C, that sort of thing. But this is telling you that you've got phase 1 down here. You've got a tiny phase here as well, phase 2. Then you've got phase 3, then phase 4. Calling them phases, not states, because states is stochastic. And there's a probability of getting down to 3 states or 4 states. But I want to pick out the dynamics of each phase. So what we do is we define the dynamics in each of these phases, looking at where the time switching is. You can also look at this cumulative probability of switching, and this clearly shows you that you've got a probability of a different phase, because this is nearly a probability of 1 to jump to this point, nearly jumped probability of 1 up to just over 3 phases. Hello.
Plots on the right, are these for this specific trajectory, or are they like across all of your trajectories?
This is one trajectory.
Just the analysis for that.
One. Yeah, got thousands to go. It's going to be a long talk.
I'm gonna go one by one.
Yeah, so we have this dynamic phasing algorithm, which tells us when things switch. Then we can pick out phases in the trajectory. So we pick out phases. Can we interpret them? Well, yes, we can. The most obvious, let me show you this movie here again. So you'll notice, there it is. If you watch this guy here, they go rapid movement. They got fast movement. These guys are peripheral kinetochores, and they undergo a fast movement toward inwards. So this is the 3 speed on this trajectory, and you can see this phase here, phase 2, corresponds to my fast movement. Okay. And then you've got these other phases. So what are those other phases? Well, this is a phase 2, the fast movement. I thought Alex would like this, that this is our orientation of the force during that phase, and it's directed inwards directly towards the mass of the kinetochores. It's not directed towards the spindle pole, it's directed towards the mass. And this is phase 3, pulling you out of that plane, probably towards the spindle pole. This is its sister. It also has 4 phases. Again, we get this fast movement here. Both of these sisters underwent fast movement, which is believed to be dynein driven. Okay, so I said probably going towards the spindle pole. That's because I've switched cell lines on you. And for a very good reason. We've developed these tools in this cell line. Wow, we're looking at data in this cell and develop these tools. So what is this cell line? Well, this cell line is MAD2 marked. Okay, this in purple is MAD2. So when this movie starts, you'll see MAD2 loading on the green kinetochores. It's loading on kinetochores, then as they become end-on attached, they start stripping off MAD2 and go green again. Okay, and this is our tracks. So the reason we were looking at this cell line is because, of course, we now have an end-on marker. It tells us when we get end-on attached. So this is our 13 and 17, the trajectories I showed you. So we've got excellent tracking in the cell line as well. And this is our MAD2 signal. Okay, this is our MAD2 signal on individual kinetochores. So you can see it rising, then falls. And at the end, they're all off. Okay, they're all off. They're all end-on attached. You get this weird one here, which seems to go through multiple phases, loading and unloading. But predominantly, you get this loading phase, and then loss. This is the noise. This is the variance of the signal in a window against the mean. You can see it's nonlinear. So to analyze this, we developed a MAD2 intensity model. This MAD2 is a noisy signal, so you have to take into account the measurement noise. So we did a phenomenological model of the measurement noise that increases with the amount of MAD2 attached. So this is the performance of our model. Again, it's Bayesian. It's got 11 parameters, 2 connect parameters, that's the loading rate and the decay rate. It's got a switch. You've got this 2 sets of hidden parameters. You've got the hidden MAD2 load. You've got a hidden state, which is either loading or unloading. And on this data, this intensity trajectory, you've got in black, it's loads and unloads and switches off. This is our switching. Because the data is noisy, of course, we're going to have quite a range switching. And this is our posterior distribution for the intensity. Track 17 is actually that really interesting one with the multiple phases. So we get switching off. This green is switching off. In between, of course, we would have switching back on again, switching back to loading. So in between here, so the interpretation is unattached. End-on attachment occurs, start losing MAD2 with dynein stripping, but it becomes detached. So it starts loading again, loads MAD2 again. And then it gets end-on attached again, detaches, but detaches down here, and so loads again, then gets end-on attached again. Okay, so we have this switching. We can determine where the switching is. We can also look at how that switching relates to our dynamic phases. So this is suggesting that you get end-on attached, then you got the fast movement, and then these other ones are in phase 4, not leaving a dynamic signature, unfortunately. So this is unusual. We normally think of a traditional picture is that the kinetochore gets laterally attached and then goes into end-on conversion. And then you go from lateral to end-on conversion. This suggests actually I've got end-on attachment followed by lateral dynein-driven motion. But of course, if you look at how much MAD2 is here, it's still very high. So you could just get there's a chance of it laterally attaching as well. And of course, although this is a MAD2 signal, dynein must also still be there. Okay? So we just think this is a chance event that we're seeing. Okay, so I haven't polished that story. That's still in progress. So we're using Bayesian techniques part of the machine learning toolbox to annotate kinetical trajectories and infer applied forces. So try and understand the mechanics. So we have switching models which can detect multiple dynamic phases in kineticals. So we can do this trajectory annotation. What we want to do is relate those dynamic phases to the spindle architecture and what's happening with the spindle, and also the checkpoint. I've shown you we've also got a MAD2 model working. And we think this gives us a powerful set of tools to start analyzing how cells go through mitosis. So we're optimizing our tools, and we hope to use these on about 1,000 trajectories. And we're putting kinetical dynamic phasing in the context of spindle checkpoints. And we want to use this to understand this collective motion of all the kineticals. So a few acknowledgements. My long-term collaborator, Andrew McCainch, generates all this beautiful data. Sarah was a postdoc who started the data generation. That's Katrina. And my postdoc, Abdulali Daniyan. He did all of the tracking. He improved the tracking algorithm, so we now get practically 100% tracking. He brought tools from radar into this, which was fun. He also did the MAD2 model, and Konstantina, she did the dynamic phasing model. So I'd just like to, I went to the Institute of Art yesterday and saw this painting. I thought, yes, I'd never come across Peter Bloom, but it said he often paints reconstruction amongst devastation. I often feel that that's how I work. Machine learning based analysis often requires rebuilding, reinventing, redoing. All those, you know, you can see behind all these methods which has failed, misled you. They weren't robust enough. Yeah, so I sometimes feel like this. So I'll leave you with that thought. Thank you.
Have you looked at the potential connections between the MAD2 signal and the stretch of the kinetochores, and how those evolve together over time?
You mean stretch of the kinetochore, meaning its shape?
Like no, I just mean like, you know, the tension, you know,
The tension, no, we haven't. No, this stuff has only started working the last 2 months. So we're still making sure they are reliable. Yeah.
Work. I have a question on the asymmetries. If you use like a drug like centrinone, do you think the asymmetries would increase, or
A drug like
centrinone, like a PLK inhibitor, PLK4 inhibitor, where you remove one of the poles?
like monastrol.
No,
Oh, you mean
centriole removal.
so have
Yeah, like
one
Eva, you know,
centriole, 3 centrioles.
yeah, the way Eva Tolish.
It should then give spindle asymmetry. So we should see, you know, you get these asymmetric spindles anyway, it's shorter this side. We should be able to, that should all come out in the analysis.
The asymmetry that you see, you know, the 75 to 25% asymmetry.
I don't know, because I would have thought if you start playing with the spindle, this spindle is stronger than the other will dominate probably.
Okay, thanks.
Question here.
So, sorry?
The static data plot you show, you have that data average from multiple tracks, and once you average from multiple tracks, you still see that nice matching between the MAP1 and the 4 phase of the kinetics. Because right now the data you show is from one particular track. I was wondering, do you still see that same 4 phases after you average from multiple tracks?
So we've done a handful of other trajectories. 4 isn't standard. Some we see 2, and it depends where you are. I've chosen peripheral ones because we know they have a fast movement. That's only about 5%. of the kinetochores undergo this fast inward movement, dynein-driven movement. The peripheral ones, so the central ones, we only get 2 phases, sometimes none. Because the dynamics in the middle is much slower. So until we've got more trajectories done, we don't really know what the statistics are like. I mean, at the moment, we're just going to focus on picking up dynamic phasing and ask whether it's towards one pole or the other. So that's our first thing we'd like to do, is know whether it's towards a pole or not, towards the mass of the kinetochores. Because this relates to the fact of where microtubules are seeded from. So if they're coming, if you're binding to laterally to a microtubule that's coming from a pole, you're going to be moving towards the pole. Whereas if you've got seeding from the kinetochores, as Alex has in his papers has been discussing, that's where you're going to get it moving more towards the network of the kinetochores.
Jing.
Beautiful work. So I want to ask, do you ever see the asymmetry kind of switch polarity, like within one pair of kinetochores trajectory?
We never looked. I mean, we were using 2-second data there, and we could fit asymmetric models. If you're asking to fit asymmetric models with a possible switch, find out when it becomes asymmetric and what form, I'd be a bit dubious of whether it would be identifiable. I mean, this is really pushing what we can get out of these individual trajectories. If you wanted to do that, I don't know. Why do you think that would happen?
Because I'm thinking about the reason, the mechanistic cause of the asymmetry. So do you think it's kind of a built-in asymmetry in the structure of the 2 kinetochores, or is it just, like a random?
Yeah, I think it's because of assembly. Fluctuations in assembly just make this K fiber stronger, that kinetochore.
Is that going to switch, like, the next time?
I know. Is it constant in time? We don't know. Good question.
Chris.
Hi, great talk. I think this is a beautiful way of finding correlation between timing of events, but I was curious, is there any hope of finding causation by putting more feedback in the model, or is this sort of the limit of complexity that these techniques can handle?
It's certainly not the limit. Certainly not the limit. Causality. So I did mention that we don't know the regulatory mechanism of the metaphase oscillations. There's a couple of models. I have wanted to fit it for years. The problem is it's semi-Markov, not Markov. And once you move out of Markov, all the beauty of MCMC and everything just gets very shaky. So I certainly believe causality can go in, yes. But as I said, here we hit semi-Markov.
All right, one more.
A beautiful talk. So what determines the number of phases like per trajectory for each trajectory? Is there a...
We don't know. We've only done a handful. I have no idea.
I see. So it's not related to the model assumptions or any...
No. We've been very careful that this ladder, I said there's 9 states it can go in. We're only touching 4 or 5. And we've done lots of model selection. One of the things I mentioned was there's this transition probability from state 1 to state 2. And on, it's a constant for all of them. And we actually fix that because you can't infer it. So we use different models with different values of that probability, and we test which one fits, and we get a whole slew of them. It's a range, which give exactly the same answers. So we're fairly confident that there's no model dependence there. But of course, what you might be referring to is the fact we've done a very simple diffusion model in there. Yes, that could be a problem. Yes.
Thank you very much. Also, one quick question. You mentioned that your model is semi-Markov. Where does the memory come from?
Oh, sorry, that's for the causality and the regulation of metaphase oscillations. I can talk to you afterwards.